Multicellular life is ubiquitous amongst eukaryotes. We are most familiar with the embryophyte plants and fungi, but they also include trabecuophyte algae, brown algae, and rhodophyte algae, all of which are just a portion of the 25 examples of multicellular eukaryotes. But between the immobile, cell-walled, and often photosynthetic or detritivorous fungi and plants, there is only one example that is both mobile and heterotrophic: the metazoans. Characterized by being multicellular, responding to environmental stimuli, reproducing sexually, consuming organic matter, and having tissues comprised of collagen IV, the kingdom of animals, alongside embryophyte plants, are the most recognizable life forms on our planet. Every vertebrate, arthropod, worm, jellyfish, placozoa, comb jelly, and even sponge shares their pedigree with one another with the mentioned plesiomorphies. Ranging from simple and delicate, such as the placozoans, sponges, and even the occasionally single-celled myoxozoa, more closely related to jellyfish, to more hardy and complex organisms, such as the vertebrates and arthropods. We too are one of the 8.7 million animal species on our planet. We are so familiar with animals, we imagine alien life forms being similar, like the humanoid aliens from various sci-fi movies and shows, or the wolf, horse, fish, and whale analogs of the Avatar series. Even Biblioridian's alien biospheres has aliens that superficially resemble polychaete worms or arthropod arachnids. But of all the diverse forms of multicellular life, why did animals only evolve once? Why is it that other colonial, heterotrophic, and mobile eukaryotes, like amoebas, did become animals? What did the ancestors of animals look like? How did our ancestors descend into the variety of forms we know today? Many, when discussing this subject, point to the likes of Ediacaran animals, such as Dixonia, Kimberella, Spurginia, and Charnia. They are the earliest fossil animals we know of, debatably, being soft-bodied, tubular, and sometimes sessile. The Ediacaran started off with the Avalon explosion, the first diversification of multicellular life. Based on how well preserved soft-bodied Vendozoan biota are, it is likely that there were very little burrowing organisms disturbing the substrate, a process known as bioturbation. The seafloor would have been dominated by microbial mats providing food for early bilaterians, as well as more obscure taxa such as Dixonia. Others resorted to filter or suspension feeding, such as Tribrachytum or Charnia. The period ended with the extinction of Ediacaran biota. The Ediacaran-Cambrian extinction event is still up for debate, but it led to the proliferation of known metazoan biota in the late Ediacaran and early Cambrian. But the Vendozoans are ambiguous about their mode of feeding, their lifestyles, or even if they're animals at all. Life during this period is split between Ediacaran biota, referred to as Vendozoans, and known metazoans. It has been argued that some Ediacaran biota could be fungi, lichens, or even an entirely different kingdom of their own. These fossils do give us clues about the evolution of animals, such as the presence of trace fossils, and known metazoans, such as Kimberella, indicates that bilaterians had already started diversifying before the Cambrian. But there's a lot more we can discover about the origins and diversification of metazoans by means of morphology, molecular phylogeny, and evolutionary developmental biology, EVO-DEVO for short, to assess the phylogeny and pedigree of animals. To start off, what were the ancestors of animals? Just like everything on our planet, animals would have started off simple, at the unicellular level. Eukaryotes are organisms characterized by their nuclear membrane withholding their genes and their membrane-bound organelles, including mitochondria and chloroplast and archaeoplastida, an endoplasmic reticulum, a Golgi apparatus, and a cytoskeleton, unlike their simpler and overall smaller prokaryotic and archaean relatives. They appeared 2.7 billion years ago, not long after the Great Oxidation Event. The taxonomy and phylogeny of protists has been debated for at least a century, with it recently being torn down and rebuilt all over again based on genetics. This is due to analogous features evolving as a result of similar selective pressures being mistaken for homologous features across eukaryotes. This is coupled with horizontal gene transfer being more common in not only protists, but also between domains in general, with examples of horizontal gene transfer between bacteria and invertebrates, like Wolbachia, genes for the biosynthesis of carotenoids and pea aphids coming from fungi, genes from algae and bacteria and bdelloid rotifers, etc. This makes evolution less of a tree and more of an interchanging web, which may be a factor that is complicating the taxonomy of eukaryotes. The taxonomy and phylogeny of eukaryotes could be a video of its own, but for now, we will only focus on where animals fit in. The protists have been classified into 6 supergroups: Archaeplastida, Excavata, Rhizaria, Chromeovolata, Amoebozoa, and Opisthoconta. Metazoans belong to the supergroup Opisthoconta. They are characterized by a single posterior flagellum and flat mitochondrial cristae. This makes animals more closely related to choanoflagellates and fungi. Their flagellums propel themselves in a single direction, as seen in the sperm of animals, the choanoflagellates, and the spores of some fungi. There is also a robust support for the opisthogonts and Amoebozoa forming a monophyletic clade, united in the assemblage Uniconta. The metazoans themselves are more closely related to the choanoflagellates than the fungi. The choanoflagellates are important when studying the evolution of multicellularity in animals. Most are solitary, free-swimming, or sessile. They are characterized by their collar composed of actin-filled microvilli. They use their single posterior flagellum to create a current, causing foreign particles and bacteria to be captured by their collar, then engulfing them through their food valve via phagocytosis. The collar and flagellum is called a collar complex. Some can also reproduce sexually via isogamous gametes, meaning the evolution of sexual reproduction actually predates the evolution of animals. Their relatedness to metazoans, similarities to primitive animal cell types, and colonial formation among a wide array of choanoflagellates makes them a keen interest among scientists to study the evolution of multicellularity. Genuses such as Choanoisa form colonies by connecting their microvilli. They invert themselves to either have a specialized feeding form or a swimming form, indicating cellular communication. The genuses Desmorella and Codosiga form connections with neighboring cells to form colonies. The genus Proterospongia, Salpingoisa, specifically Salpingoisa rosetta, alternates between multiple solitary and colonial forms, either becoming slow-swimming, fast-swimming, or thecate forms, or forming colonies of tightly packed spheres that range from 4 to 50 cells, or chain colonies. All formed via incomplete cellular division and connected via mix of intercellular bridges and extracellular matrix and filopodia. The species alternates depending on environmental conditions. Spherical rosette colonies form as a result of the introduction of the bacteria Allergoriphagus machipongonensis. And chain-like colonies form under rapid growing conditions. Both colonial forms are derived from a solitary slow-swimming form. Salpingoisa rosetta, for the reasons provided, is a keen interest among scientists for their cellular differentiation. Based on the multitude of examples of colonial formation, it has been proposed that the formation of colonies would have been ancestral to Holozoa, but it is less parsimonious to propose that colonial formation is convergent in choanoflagellates. What is agreed upon is that the ancestral Holozoan would have resembled the modern choanoflagellate. Furthermore, it already appears as though the choanoflagellates have the basis for the 2 major steps in animal evolution: multicellularity and cellular specialization. Speaking of animals, the metazoans are grouped under 5 distinct groups: Porifera, Ctenophora, Placozoa, Cnidaria, and Bilateria. The bilaterians are an infrakingdom of animals, characterized by their bilateral symmetry and their triploblasty, meaning they have 3 tissue layers: the endoderm, the mesoderm, and the exoderm. This is unlike former eumetazoans, which are diploblastic, bearing only an endoderm and an exoderm. Bilaterians are one of the most diverse animals on our planet, including arthropods, annelids, mollusks, platyhelminthes, chordates, even the radially symmetrical echinoderms, which lost their bilateral symmetry. Cnidarians are a phylum of animals characterized by their tentacles with stinging nematocysts, radial symmetry, and have alternating sexual and asexual phases. They include the sea anemones, jellyfish, siphonophores, and even the corals. The placozoans are considered to be the most simple animals on the planet, with the genus Trichoblax bearing only 4 to 6 cell types, and are shaped like pancakes or flat amoebas. They are still considered diploblasts, digesting food using their ventral epithelium to form a digestive cavity between it and the substrate. Both epithelial layers bear ciliated cells, the squamosal and cylinder cells, and the fiber cells within their mesoglea act as both nerves and muscles, allowing placozoans to contract and move, but also using neuropeptides for chemical signaling. The ctenophores are the comb jellies. They are characterized by their coloblasts for feeding and have biradial symmetry, and bear temporary anal cavities. They will be important for discussing the phylogenetic positions of all other metazoan groups. Finally, we have the poriferans, or more ubiquitously known as the sponges. They are the most primitive animals, having only one tissue layer and perform intracellular digestion, meaning digestion isn't aided by enzymes or acids, but rather takes place within the cells. They suck water through their pores called ostium, filter bacteria and particles, then pass it through their gaping osculum. Their outer surface is composed of cells called pinacocytes. Their role is essentially to cover the sponge and protect their mesohyl. Their mesohyl is a jelly-like matrix containing archaeocytes, which transport nutrients throughout the sponge and act as stem cells, and sclerocytes, which produce the sponge's skeleton. Finally, inside the sponge are choanocytes, which are cells that bear a collar complex to create a water current and capture bacteria and foreign particles. The choanocytes have always been noted to have a striking similarity to the choanoflagellates, further uniting these two groups. Because of their primitive body plan and cellular anatomy, the sponges have been of particular interest among scientists to understand the evolution of animals from choanoflagellate-like ancestors. However, the relations between these metazoan groups have been largely contested for well over a century. Starting at the base of Metazoa, it was always agreed upon that sponges were the most basal, forming a sister group to all other metazoans. The similarities between sponge choanocytes and choanoflagellates was first noted by James Clark in 1866. He then correctly concluded that sponges evolved from a choanoflagellate-like ancestor. It was then thought by Haeckel that they weren't even animals but protists, then eumetazoans by Schultz based on their mode of spermatogenesis, until genetics proved sponges as a group of animals and choanoflagellates as a sister group. But the evolution from unicells to sponges had been left up to debate. Flagellated cells forming colonial spheres have been known since Volvox was described in 1677. Their similarities to the blastrula stage of embryonic development in animals, and their abundance in colonial cells, makes them a plausible candidate from unicells to sponges. Ernst Haeckel was the first to reconstruct the evolution of animals from unicells. He was convinced the ancestors of animals were amoebas, based on the embryonic development of sponges, and a species he was convinced to be the missing link, Megasphaera planula. He proposed the ancestors of animals started off as an amoeba-like Citaea, which evolved into colonial spheres called Amorea. Then it became ciliated, becoming a free-floating Blastea. Ernst Haeckel is credited for coining the term gastrulation, the stage of embryonic development where the blastrula invenates, forming the archenentron, which is then derived into a gut. So he was the first to propose a Gastrea-like ancestor. Haeckel's hypothesis for the evolution of animals never gained wide acceptance for unknown reasons, but it is likely due to competition with a more accurate hypothesis, ironically inspired by Haeckel's. Russian biologist Elie Mechnikov, influenced by Haeckel's amoeba-based hypothesis, first proposed a choanoflagellate-like ancestor based on similarities to sponge choanocytes, better explaining the presence of a flagellum as being homologous as opposed to convergent. It has been hypothesized that the ancestral animal would have evolved from a rosette-like ancestor. An established epithelium via tight cell contacts led to a choanoblastea. The establishment of an epithelium leads to better cellular communication and nutrient transfer, allowing more specialized non-feeding cells, leading to an advanced choanoblastea. So it is not unlikely that a choanoblastea evolved to sit on non-ciliated cells, become larger, lithotrophic larvae, than furrows that were derived into internal canals. But the idea of sponges being a sister group to Eumetazoans has been contested by the ctenophore first hypothesis. Previously, the comb jellies were grouped with the cnidarians within Coelentatra, based on their overall similarities to one another and the presence of nerves and muscles in both clades. Recently, however, it has been proposed that the ctenophores actually came first based on molecular phylogeny. This would indicate that either sponges lost nerves and muscles just to re-evolve in Planulozoa, or the evolution of nerves and muscles are convergent in Planulozoa and ctenophores. This would significantly reshape how we reconstruct the taxonomy and evolution of metazoans. But it turns out the ctenophore first hypothesis was more of a result of poor gene taxon sampling, thus making the hypothesis dubious. But the ctenophores are still no longer considered related to cnidarians for reasons we'll get into, thus making Coelentatra outdated. So the consensus on porphyrians over the last couple centuries has made full circles, going from sponges being animals, then protists, then a sister group to other animals, Eumetazoans, then back to the first metazoans. Now that we've discussed the base of metazoa, let's talk about Eumetazoa. Weirdly, for over half the 20th century, it was thought by most that animals were polyphyletic. It was first proposed by Seville Kent in the late 19th century, who provided 2, maybe even 3 origins of animals, with sponges being derived from choanoflagellates, bilaterians were perhaps descended from ciliates, and possibly cnidarians from amoebas. Kent and followers of his theory noted similarities between ciliates and acoelomates alongside planula and trochophore larvae. The anatomy of acoelomate worms were very hard to determine due to their simplicity, with their lack of coelom, oral and aboral end, and even a proper gut. It was even thought at one point that they weren't even multicellular, but multinucleated. The ciliates under this light would be very similar to acoelomates, bearing a primitive mouth called a cytosome, a digestive valcule, cilia, a micro and macronucleus, and pulsatile valcules. The ciliate hypothesis would be largely ignored upon its proposal, then brought back independently in the early 20th century. Surprisingly, this hypothesis sustained support until electron microscopy in the 1960s proved acoelomates were multicellular, not multinucleated. In addition came molecular phylogeny, which initially supported the ciliate hypothesis and a polyphyletic origin of animals, but later went against it, then, as mentioned, favored the choanoblastea hypothesis. Unfortunately, the ciliate hypothesis and a polyphyletic origin of animals was still thought of as likely into the 80s and 90s, even being supported by respected biologists such as Stephen Jay Gould. The consensus now is that Metazoa forms a monophyletic group, with sponges being outside of Eumetazoa. Eumetazoa is characterized by their extracellular digestion and undergo gastrulation, giving them 2 epithelial tissue layers. Reconstructing the taxonomy of Eumetazoa and the ancestral state has brought up just as many questions. There was initially a variety of hypotheses reconstructing the origins of Eumetazoans and how it would dictate Eumetazoan phylogeny, such as the Placozoans, representing the ancestral Eumetazoa due to their simplicity. But this has been disputed based on a Placozoa-like stage not being expressed during embryonic development, and due to the presence of para-Hox genes. Hox genes code for body polarity and formation, such as where your limbs and head go. They were first discovered in the 80s in mice and fruit flies. This suggests deuterostomes and protostomes descended from an ancestor like a Eumetazoan. Para-Hox genes, related to Hox genes, have been found in cnidarians and Placozoans, but are lacking in ctenophores and sponges. So within Eumetazoa, ctenophores are placed as the earliest diverging lineage, and Bilateria, cnidaria, and Placozoa unite to form the group Parahoxozoa. So what does this indicate for the ancestral Eumetazoan? Gastrulation is important in the evolution of Eumetazoans. It's what helped early animals rise from hollow multicellular balls to have complex organs and tissue layers. So, the most accepted theories reconstructing the ancestral Eumetazoan are the terminal addition theory and the intercalation theory. The terminal addition theory proposes Eumetazoans started off as a holoplanktonic gastrea. Then, a benthic adult stage was added later in Planulozoa. The intercalation theory proposes Eumetazoans started off as a benthic creeping adult, possibly even bilateral. Then, a holoplanktonic larval stage was added later. Both of these theories were based on larval development being present in most bilaterians and cnidarians, such as the trochophore larvae of spiralian protostomes, the diplural larvae of Ambulocruria, and the planula larvae of cnidarians. Juvenile ctenophores look practically like adults. But the intercalation theory suffers from the fact that an apical organ, as well as ciliary bands, are present in most larval Eumetazoans. In addition, the presence of para-Hox genes in Parahoxozoa, but not ctenophora, suggests either ctenophores lost their para-Hox genes, and ciliated larvae with apical organs evolved convergently in bilaterians, or the ancestral Eumetazoan was actually a holoplanktonic gastrea that already had an apical organ with cilia. Then, a creeping adult stage was added later. Related to the terminal addition theory, it has been proposed that the Eumetazoan gastrea could have descended from larval sponges. Because, as mentioned earlier, some demosponges do undergo gastrulation. But it could also be more likely that gastrulation is convergent in sponges and Eumetazoans, as opposed to homologous. The Parahoxozoans have robust support based on molecular phylogeny. The consensus was that Placozoans were the earliest diverging Parahoxozoans, and Bilateria and cnidaria had sister relations, forming the group Planulozoa. But there's equal support for the sister relations between cnidaria and Placozoa, indicating that Placozoans either lost adaptations, such as nerves and muscles, or, more unlikely, Placozoans represent the ancestral state of Parahoxozoa. I will let you decide which theory you support most, but for the sake of this video, I find the Planulozoa theory more likely. Regarding Planulozoa, it has been debated whether the ancestral Planulozoan was radially, or at least semi-bilaterally symmetrical. The cnidarians have always been considered radially symmetrical, being akin to cylinders. But some cnidariacean enemies do exhibit bilateral symmetry to some degree, such as the model species Eumetazoa vectensis. This brings up whether bilateral symmetry is homologous or convergent in Planulozoa. There is a support for the homology of bilateral symmetry, with the identification of 5 Hox genes in Eumetazoa vectensis. This indicates the ancestral Planulozoan was bilaterally, or semi-bilaterally symmetrical for unidirectional movement. The evolution of cnidarians was a result of a sedentary lifestyle, losing most of their bilateral symmetry and aided by stinging nematocysts. Bilaterians evolved as a result of a further adaptation to a benthic feeding lifestyle. Now we have a clear understanding of how animals evolved. Before the armored, carnivorous, actively swimming or burrowing animals of the Cambrian, or the creeping, soft-bodied, microbial, or filter-feeding Vendozoans of the Ediacaran, animals started off at the unicellular level during the Cryogenian period. Animals trace their pedigree to free-swimming Choanoflagellate-like ancestors, feeding on bacteria and foreign particles by means of their flagellum, drawing food into the collar complex. They likely participated in rosette-like colonies by means of incomplete cellular division and alternated between multiple cell types depending on environmental conditions. The evolution of an established epithelium via intercellular bridges composed of filopodia and an extracellular matrix composed of collagen IV allowed for more advanced non-feeding cell types. Now we have the first animal, an advanced Choanoblastea. From this ancestor branched off a lineage that adapted to sedentary filter feeding. Specialized non-flagellated cells allow the Choanoblastea to adhere to rocks, and amoeba-like cells transporting nutrients and acting as stem cells led to pre-sponges. Then furrows on its surface for more food consumption led to internal canals, then pinacocytes covered the outer surface for protection. The establishment of ostia and an osculum led to asyncoid porphyrians. Another branch, either from the advanced Choanoblastea or larval sponges, adapted a pelagic holoplanktonic lifestyle. This allowed animals to pass from hollow sphere cells via gastrulation, forming the archenteron for extracellular digestion. This was facilitated with the reduction of the collar complex and an apical organ for lysensation, leading to a gastrea. The evolution of extracellular digestion allowed animals to digest larger particles, thus expanding their range of ecological niches beyond bacterial filter feeding. The advantages of gastrulation and extracellular digestion allowed for the diversification of Eumetazoa. The first diverging branch adapted to a sedentary filter-feeding lifestyle, aided by sticky colloblasts, nerves, and epithelial muscles, leading to the ctenophores. The next monophyletic branch likely adapted to benthic feeding, aided by parahox genes for body polarity. The earliest branch expanded their blastopore so their endoderm performed digestion with the substrate itself. Then they evolved fiber cells in their mesoglea to change its shape and perform neuropeptide signals, leading to the placozoans. Following the parahoxozoans, the gastrea ancestor either became creeping and semi-bilateral, with the larvae maintaining cilia and an apical organ, or they remained as a neurogastrea. This planulozoan ancestor branched into two lineages. If the former is true based on Hox genes, the ancestral cnidarian lost most of their bilateral symmetry in favor for sedentary feeding, aided by nematocysts. Their gastrea-like larvae were retained, known as planula larvae. Finally, the other lineage adapted further to benthic deposit feeding, evolving full bilateral symmetry for unidirectional movement. This adaptation allowed for the most mobility, pre-adapting animals for a wide variety of niches and morphological complexity not tapped by most cnidarians, placozoans, and the ctenophores. Burrowing, active swimming, predatory, even crawling animals owes its success to the establishment of an oral aboral and dorsal ventral axis from its eubilaterian ancestors. There, they would branch into protostomia as an adaptation to benthic deposit feeding, and deuterostomia as an adaptation to benthic suspension feeding. The taxonomy and phylogeny of bilaterians has its own history of discovery and research, but this would be a topic for a later video. In conclusion, the evolution of metazoans was a result of a simple, colonial rosette-like ancestor being modified for intricate complexity as it adapted to mobile, sedentary, or free-floating lifestyles in pelagic or benthic environments. There's still plenty we don't know, such as where the Vendozoan biota fit in, but under this light they may be easier to understand. Dickinsonia, as an example, has been interpreted as a placozoan, and Charnia has a significant resemblance to soft corals. This is not to say that they are equivalent, but they can be seen as having similar lifestyles. With what we know now, we can use this to understand not only how animals evolved on Earth, but you could also imagine how alternative animals could evolve on alien planets. We could also use the history of our understanding of metazoans to always be skeptical, but also not dogmatic. Being open to new theories and ideas is what pushes old and outdated ones out, just as long as it's based on facts and evidence first. With this being said, this concludes the longest video on my darn channel. So far, at least. I'd be surprised if anyone made it towards the end, especially nowadays with YouTube Shorts and TikTok. I'm not feeding into that. I think you skipped towards the end. I'm not gonna allow that. I can make it longer if you want. Is your attention span longer yet? Okay, that's about it. I hope to see you soon. I'll see you in the amount of time it took for metazoans to evolve. Bye.